Good evening everyone. In this video I'm going to talk about the short notes on the Fundus Autofluorescence first chapter by Forrester. You don't need to go through the entire 40 minutes long video until and unless you have a lot of time, and I don't think being a doctor you have a lot of time and you have to use the best use of time. If you want to destroy anyone's life, just waste that person's time, and that's what these people are doing. I don't want to get into that thing, but just save your time. You don't need to go through that 40 minutes long video. You can directly jump on this video. So in this video I'm going to talk about the definition of the autofluorescence and the fluorophores in the human body, the causes of accumulation of them. Then I'm going to enlist the diseases and conditions in which they are accumulated in the body, and then the names of these fluorophores, mainly two, lipofuscin and steroid, and the fate of cell body in which these fluorophores are accumulated. So first definition, autofluorescence. It occurs at the level of molecular level or atomic level or electron level. This autofluorescence happens. So what is this? When a molecule or atom or electron, it absorbs a light wavelength of a relatively shorter wavelength, a light of a shorter wavelength, it gets excited, and when it comes back to the normal position or resting position, it emits out a light of a wavelength that is of longer wavelength than the wavelength that it was incident upon it. So in human body, autofluorescence property is exhibited by macrophages due to accumulation of two fluorophore types, lipofuscin and steroid. Now lipofuscin, it is accumulated under physiological process and even in certain diseases. For physiological process, for example, aging process, and certain diseases related to aging process, like in old age these diseases are seen, and some are hereditary or genetic diseases. Then second is steroid. Actually it is accumulated in the human cells only in certain pathological conditions, not in physiological conditions. For example, diseases, neuronal degenerative diseases. Then the disease is called as neuronal steroid lipofuscinosis. Now what are the causes of accumulation of these fluorophores, which are lipofuscin and steroid, in our human body? So first mechanism is autophagy, cellular stress and lysosomal dysfunction, that we will go through little bit in detail, not much, little bit in detail later on. Then there are certain conditions in which lipofuscin is accumulated in the human body or steroid is accumulated in the human body. So there is a table 1.1 here given in this book. Occurrences of lipofuscin or steroid: aging, age-related macular degeneration, neuronal steroid lipofuscinosis, mucolipidosis 4, mucopolysaccharidosis 3, Sanfilippo disease, Alzheimer's disease, retinitis pigmentosa, cone and rod dystrophy, X-linked retinoschisis, Leber congenital amaurosis, pattern dystrophy, Best disease, Stargardt disease, maternal inherited diabetes and deafness, choroideremia, osteopetrosis with neuronal storage disease, adult onset glycogen storage disease type 2, macular ABCA4 disease, Wilson's disease, Crohn's disease, choroidal tumors. Once again, I would like to enlist the conditions in which there is accumulation of the lipofuscin or steroid: aging, age-related macular degeneration, neuronal steroid lipofuscinosis, mucolipidosis 4, mucopolysaccharidosis 3, Sanfilippo disease, Alzheimer's disease, retinitis pigmentosa, cone and cone rod dystrophy, X-linked retinoschisis, Leber congenital amaurosis, pattern dystrophy, Best disease, Stargardt disease, maternal inherited diabetes and deafness, choroideremia, osteopetrosis with neuronal storage disease, adult onset glycogen storage disease type 2, macular ABCA4 disease, Wilson's disease, Crohn's disease, and choroidal tumors. Now what is the composition of lipofuscin and steroid? There is a comparative table in this book, table 1.2. Lipofuscin, location primarily lysosome, steroid, location primarily lysosome. Now spectral properties of lipofuscin: it absorbs or it gets excited at 320 to 460 nanometer wavelength of light, and steroid is excited at 320 to 460 nanometer wavelength of light. And lipofuscin and steroid both, they have similar emission properties. They emit the light wavelength of 460 to 630 nanometer. So the emitted light wavelength is 460 to 630 nanometer. Both lipofuscin and steroid, they have both excitation and emission spectral properties which are similar. So excitation is 320 to 460 nanometer, and emission is 460 to 630 nanometer, longer wavelength. So what is the chemical composition of lipofuscin and steroid? So first, chemical composition of lipofuscin. It's made up of proteins, lipids, carbohydrates and metals. The protein is A-beta-PP, that is amyloid or amyloid beta precursor protein, heterogeneous mixture. Then lipid part, triglycerides, cholesterol, phospholipids, free fatty acids. These are the lipids. Carbohydrates, heterogeneous mixture. Then metals, iron, copper, aluminium, zinc, Mn. Mn is manganese, but I don't know, here they have written magnesium in the paragraph, and here they have written Mn, that means manganese, and magnesium is Mg, right? So Ca is calcium. So proteins, protein part is around 30 to 58%, and lipid part is around 19 to 51%, almost similar, but protein part is more, 30 to 58% in most of the situations. Lipid part is 90 to 51%. So that was about the composition of the lipofuscin. Now what is the composition of steroid? Steroid protein component, subunit C mitochondria ATPase, or saposins A and D, and amyloid beta precursor protein, and lipids, phosphorylated dolichols, phospholipids, neutral lipids, then carbohydrates, dolicol linked oligosaccharides, and metals. In steroid it is predominantly iron. Now staining characteristics of lipofuscin: Sudan black B present, periodic sheaf base, yes. Then lactin, concanavalin A, and ultrastructures GRODS. GRODS means granular osmophilic deposits. GROD means granular osmophilic deposits. Then coming to steroid, stain properties are similar. Sudan black B present, yes. Periodic sheaf base, yes. Then lactin, again concanavalin A, and agglutinin, similar but additional agglutinin here. And ultrastructure, again GRODS, fingerprint and curvilinear in case of the steroid. So GRODS means granular osmophilic deposits. Granular osmophilic deposits, GRODS. So this was all about the composition of lipofuscin and steroid and the conditions in which their accumulation occurs. Now what is the effect of accumulation of lipofuscin on the cell body? What happens when the accumulated, it goes on building up inside the cell? What happens? This lipofuscin accumulation, it triggers the apoptotic cascade to initiate the cell death. Eventually what happens? It will stimulate the, or it will trigger the apoptotic cascade. Apoptotic means programmed cell death, and it will initiate the cell death. So cell death means decrease in the cell number in the tissue, and the tissue or the organ function, it declines. So little bit in detail about the causes of accumulation of the lipofuscin. There are three main causes: lysosomal dysfunction, autophagy, and then cellular stress. Now lysosomal dysfunction. Lysosomal dysfunction, it occurs in case of the mutation senescence or chloroquine. What happens in these conditions? Senescence means aging, whether there is a mutation, aging, or chloroquine toxicity or chloroquine. What these things do, they will decrease the function of the lysosomal enzymes involved in the catabolism or digestion or renewal of the lipofuscin and the cellular waste products. Then what will happen? There will be accumulation of the lipofuscin. So what chloroquine does is it alters or raises it raises the lysosomal pH. Raises the pH means it will make it more alkaline, and that will lead to the accumulation of the lipofuscin in hepatocytes and brain cells. Second mechanism for accumulation of the lipofuscin is autophagy. Autophagy means, it's a Greek word which means self-eating. Self-eating, it can occur in physiological conditions or pathological conditions. Physiological conditions means aging, exercise. Pathological conditions in certain diseases and in, for example, cancer, infections. Now what happens in autophagy? The cell eats its own contents. It involves delivering of cytoplasmic waste materials or bacteria or viruses to the lysosomes for recycling or clearance of them respectively. For example, what happens? What are the steps involved in autophagy? First is phagophore formation, then autophagosome formation, and then autophagolysosome formation. So phagophore, it is made up of the invagination of plasma membrane or mitochondrial membrane. Then there is autophagosome formation, and then fusion of the autophagosome with lysosome to form the autophagolysosome, and they undergo degradation because of the enzymes in the lysosomes. So this is how the autophagy takes place, to clear the cellular debris or bacteria or viruses or the proteins and amino acids for their renewal, and lipids for their renewal. So third mechanism is cellular stress. What happens in cellular stress is, in normal conditions there is generation of reactive oxygen species always and always. In mitochondria, whenever there is ATP generation, what happens? There is oxidation of certain things, and reactive species generation, hydrogen peroxide, superoxide radical, and hydroxyl radical. These radicals are very harmful. They are very unstable. They will just go and they will oxidize the lipids and proteins in the cytosol, in the cytoplasm, in the cell membrane, and they will cause the cell damage. So we don't want these reactive oxygen species. So there is a protective mechanism for that inside our body, and these enzymes include catalyzed glutathione peroxidase and superoxide dismutase. But actually this is not enough. This doesn't protect our cells or body from these reactive oxygen species completely. Some of these species, they escape this mechanism and they keep on damaging our cells. But this is under balance, and this balance, and as we age, this balance is more on the destructive side. This is lost as we senesce, as we age. So that was all about the lipofuscin, autofluorescence. And one thing I forgot to mention is this autofluorescence in the lipofuscin. They observed that it is because of either peroxidation of lipids, or there is a fluorophore substance in this lipofuscin, that is pyridinium bisretinoid, or this is a complex of multiple fluorophores which is responsible for the fluorescence of the lipofuscin. We don't need to go so much in detail in that. We just remember that this fundus autofluorescence is because of, especially as an ophthalmologist, we should remember that it is because of the lipofuscin molecule, and it gets accumulated. The primary location of it is lysosome, and in the second chapter we'll see its connection to the retinal pigment epithelium, and it has autofluorescence and fundus autofluorescence. And there are certain conditions which will in which there is a lipofuscin accumulation. I have already enlisted these conditions. You can go through them if you want to just by heart them, and you don't need to by heart them, as we will go through these chapters in this book. We will remember them seeing the pictures of them, so you don't need to just by heart blindly right now. You just go through them once, and then we will just keep on reading these chapters fast, because I didn't get a nice video on fundus autofluorescence on YouTube. So I decided to make one nice video on fundus autofluorescence. This is just notes part, and later on at last I'll just combine certain pictures, and in short I'll be talking about fundus autofluorescence, and that video won't be more than 1 hour, because 1 hour or 1.5 hours, because I'm going to combine all the pictures and mechanism of fundus autofluorescence. as an ophthalmologist, you don't need to know everything related to the pathology and biochemistry of this fluorescence and even the physics of the fluorescence. A little bit that has the practical significance, the theory or related to it, you need to know for the better understanding. And this much is too much for you, but I do have time right now, so I decided to read about it, because I didn't read much about it. I just knew about the autofluorescence and its practical aspects and certain conditions and certain photographs I knew. So I decided to study those chapters in detail which I didn't study that much in detail during the exam. So I chose this chapter, Fundus autofluorescence, and I feel sad that I have never seen such machines in government institutes, and that's why I don't want to work in the government institutes, because there are no machines. Even if there are machines, there is a lot of politics, and the kind of verbal treatment, the kind of politics these institutes have, that's pathetic. Even the charity hospitals, the quality of the treatment is very bad. Even the quality verbal treatment and behavioral treatment and the politics is too much. And charity is nothing but they just want you to, you know, they want free laborers in the name of charity. It's not a charity, it's actually a slavery on behalf of the doctors. Like, if you want to learn something and if you're working and if you don't have, if it is like a rich man's kid or rich man's child, it's fine, like they can just work even free of cost, but not the people like me who need money and who need a qualitative treatment and a respective environment, a healthy environment, not abusive environment, not violent environment. I just hate abusive and violent environment. I don't get abusive and violent primarily. If someone behaves with me like that, then I don't have any option, because if you just keep on being silent, then people take advantage of that thing. So if you want to know about advanced machines, or you want to learn or get advanced skills, there is no option but to join a private institute, a good private institute, a good teacher who is respect worthy, who is honest, moral, who earns in a noble way, and is generous enough to transfer his or her knowledge and skills onto you, and is not jealous to make you do or make you perform better than them also. So such kinds of teachers I have not seen, honestly I have not seen. But you have to work, you have to earn. So I'm just in search of a retina surgeon, a good surgeon that will who will be very generous to transfer his or her knowledge and skills onto me. So I'm just looking forward to work with a good, successful and intelligent, moral, noble, and humble. I don't like abusive and arrogant people. I don't like to work with such people, absolutely not. I just respect humbleness and morality and not arrogance. Even if that person is skillful and knowledgeable, I don't give respect to such people. Yeah, so that was about the Fundus autofluorescence. I hope you get to I hope I get to see the machine, and actually I work with this machine in real life, because I want to see even FFA in real life. I want to do all these procedures, and in government sector you will never get to do such advanced things, and there are no advanced machines. So I'm looking forward to work in a very nice private institute where I can just I am gaining knowledge about these advanced things, and I hope in near future, in coming future, I'll be able to handle these machines. I'll be working with all these machines, and my knowledge will be actually in practical utility for the benefit of me and for the benefit of the patients, mainly benefit of the patients. And when the patients are benefited, obviously I will be benefited. Bye everyone.